thanks so much for inviting me to be here today. I'm excited to tell you about some of the work that's been going on in our lab. I've been here at UCI about going on 2 years now, so it's enough time that I've started to grow a little bit of a lab group who are with me here today. So they're going to be walking around and introducing themselves soon. Wave, you guys. Right there. What I'd like to do before we jump into talking about some of the science is give you a little bit of a primer on some of the more complex physical chemical processes that I think about in the ocean. So I'm an oceanographer, but I have kind of a untraditional background. I started out being really interested in engineering and botany, and as a kid I always thought I'd end up in the rainforest studying land plants. And if you come to my office in Crawl Hall, you'll see that that still carries on to this day. It looks like a rainforest in my office. But I had a really serendipitous experience when I was about 18 years old. I did a field study class in the Sea of Cortez, which is the Gulf of California. And my plan was to study island biogeography. So that's like the study of why plants grow in certain areas on all of these different islands in the Sea of Cortez. And what I didn't expect, and what I ended up really falling in love with, was the fact that we were living on this boat. Down here on the bottom, you can see the little boat that we lived on for 2 weeks. And some of the instructors were oceanographers. And I learned about the ocean during that time, and it blew my mind. One of the first things that I learned about was that the ocean is not just this big homogeneous bathtub. It has different water masses, and they have different chemical and physical processes. And whether or not they mix determines what nutrients are available for phytoplankton, and that in turn determines what higher plants and animals can grow in that area as well. So the fact that our coast is so different from the East Coast, for example, is directly an effect of these chemical and physical processes. So here's an example of kind of a global scale circulation pattern that's driven by density gradients in the ocean. And the density is just basically, if you have a certain volume of water, what's its mass? The more salt that's in that volume of water, the heavier it's going to be. So we say that's denser water. That tends to sink. And we'll do an experiment to demonstrate that. But the way that this process actually gets driven is that you have warm, shallow surface currents that are skating along the top of the ocean, and they go up towards the poles, and they start getting colder and colder and denser, and in some cases saltier and saltier, and they sink. And as they sink, they push the deep water out of the way, and they create this conveyor belt. And they push the water around the ocean. So this is how the nutrients are getting distributed that the phytoplankton I study are trying to use. Density is driven by two principal ideas in seawater, the temperature and the salinity. And again, that's just because how warm the water is determines how much it expands and contracts. So that's going to change the mass per volume ratio, which is density. And the amount of salt that's in there is also going to affect it, because the salt has mass. So if you put salt in the water, it becomes more dense. So I have on each of your tables a couple of sets of cups. And one of the cups has an S for salt water, and one has an F for fresh water. And so before we go into all the other science and implications, let's just get a hands-on feeling for what it means to have stratification in a cup, so that we can think a little more about what it means to have stratification in the ocean. So what I'd like everybody to do is just take your freshwater and saltwater cups and let them sit on your table so that they're nice and still, not shaking or sloshing around. And everybody should have a cup of ice. Add an equal amount of ice to both cups. I'm thinking somewhere on the order of maybe 4 ice cubes per cup is going to be fine for what you guys have. Just make sure it's a similar amount for each one. Try to add it gently so it doesn't, disturb the water too much. Okay, so once everybody's added their ice, we're going to give a couple of minutes just to let the ice melt. But as you're sitting there watching your ice melt, which is fascinating, think about some of the questions I'm about to ask you, like which cup do you think the ice is going to melt faster in? You can talk to, some people are saying fresh, I hear some people saying salt. Talk to the people next to you and tell them why you think that it's the fresh water or the salt water that's going to melt faster. Okay, who has an idea about which one is going to melt faster? Let's do a show of hands. Who thinks that the saltwater ice is going to melt faster? Okay, about half of us. And who thinks that the freshwater cup is going to melt faster? A little less than the other half. Okay. So the people who are on the saltwater side of things, why do you think that the saltwater is going to melt it faster? Yeah, so there's a freezing point depression that happens when you add salt to something. And the way that it melts the ice on the sidewalk is that the ice is in a dynamic equilibrium. It melts a little bit and it refreezes. But if there's salt there, it prevents refreezing. So the net effect is that you start to get melting. But I'm curious about the people who said freshwater. Who said freshwater, and what's your argument? Yes?
It's a larger volume, so the volume would probably make it go faster.
What do you mean by a larger volume?
On our table, the salt water had a larger volume of water.
Oh, okay.
Freshwater had a larger volume of water.
Okay, well, they should have had the same amount, but so that shouldn't have too much of an effect, hopefully, but that's a good guess. Anybody else want to take one more stab? Yeah. Interesting hypothesis. Okay, let's take our food coloring and add a few drops to each of the cups and see who was right. What do you notice about the way that the dye behaves when you add it to the freshwater cup versus the saltwater cup? Okay, so now, by a show of hands, once you've had a chance to look at it, which cup appears to be melting faster, the saltwater or the freshwater? Let's see, who says saltwater? Yeah, who says fresh? Yeah, so it's stratification. The fact that the fresh water is less dense and it sits on top, you can see that because of the way that the food coloring is, right? So that cold ice gets trapped in the surface and it melts more slowly because it's surrounded by cold water. Whereas in the freshwater cup, there's no density difference, so as soon as the ice melts, it can sink. That's exactly what happens in the ocean. So when we have mixing in the ocean, we can homogenize all of the different layers, and that distributes the nutrients for the phytoplankton to use. But when we have stratification, everything's stuck in the top, and it tends to get really, really sparse up there in terms of nutrients. It's kind of like a desert. There's just not a lot of stuff. So we're going to talk about a couple studies that get into that today. We talked a little bit about which cup had the fastest melting, and I think everybody was a little bit surprised. Full disclosure, I was too the first time I did that experiment. But the reason that we care about stratification in the ocean is because it plays into all sorts of questions that we're looking at in modern science. And they tie into climate change because through warming, we expect to have greater stratification of the oceans. And that in turn will change nutrient availability patterns. And that affects who grows where and why, which is what I study. Which phytoplankton are growing in which locations and why do they grow there. So today I'm going to talk to you about two major studies that both have a little bit to do with the way that physical and chemical processes in the ocean affect the phytoplankton and how humans are playing a role in that. But I'll start with a really basic introduction to some oceanography in case you haven't been exposed to it yet. My first exposure, like I said, was when I was 18 in that Sea of Cortez field trip. But it is a fascinating topic. So I'm in the Earth System Science Department here at UCI. Most of my colleagues, when they think about the Earth, are thinking about the beautiful blue marble. But when I think about the Earth, it's a little bit of a different picture, because I'm thinking about the half of the planet that is water, and then half of the other half is also water. So it's a tremendous surface area over which biogeochemistry can occur, and through which humans can have an impact. The particular process that I study in great detail is photosynthesis. And photosynthesis in the ocean is very similar to photosynthesis on land. Basically what happens is that no matter who you are doing photosynthesis, you need a source of light, and that's typically the sun. And then carbon dioxide, which either comes from the air or it's dissolved in the water if you're a phytoplankton cell. Together with nutrients and some water, they combine to make new cells, and then they generate oxygen as a waste product. And that's the stuff we breathe, so we're happy about that. The reason that this matters on an ecological scale is because those little phytoplankton are acting as a pump to get nutrients into the food web. So you think about it, they're taking stuff like nitrates and phosphates that are useless to us, and their bodies make them into usable forms, like proteins and DNA that we can then digest and make as part of our cells. As they do that, they're also supporting these really important marine mammal communities that are very charismatic and that people like to come to our coast to visit, as well as biodiversity in coral reef ecosystems, and even economically important fisheries. And now I think Definitely in my case, I grew up thinking a lot about land plants and thinking about global primary productivity, which is just the amount of photosynthetic stuff that's available for animals to eat. And if you think about it, most people would probably say that most of the carbon biomass in the planet is in the form of trees. And if you thought that, you would be correct. It's about 99%. So if you weighed up all of the carbon biomass from photosynthesis in the world, about 99% of it is tied up in these big trees. But the phytoplankton are growing really, really fast. And they're covering a vast area of the Earth's surface, down to about 200 meters, where it's very dim. They're getting eaten all the time. So the difference is that when a tree makes some carbon, it stores it in its stem or its leaf and it sticks around. When a phytoplankton cell makes new carbon, it's putting it directly into the food web. So they're actually doing the same amount of photosynthesis, and they're fixing the same amount of carbon. What that also means is that they're all producing the same amount of oxygen, so every second breath you take came from these algae in the ocean. So today I'm going to talk about some coastal environments. And coastal environments are different from the open ocean because they've got a lot more nutrient sources. They've got these rivers that bring in urban pollution and nutrient sources. Even though they only cover about 15% of the ocean surface, they're responsible for about half of the productivity that the ocean does. And so they're supporting a really high biodiversity of phytoplankton, and those guys in turn support really diverse fish populations, which are good for our economy and for our well-being. At the same time, they're really susceptible to bad things like harmful algal blooms because of the way that we alter the coastal environment. And there's a lot of social interests tied up in coastal environments. People are spiritually tied to these coastal waters. So there's a number of reasons to try to understand how future changes in our planet are going to manifest in these systems. Southern California in particular is a really good example of a place where we derive our livelihoods from the ocean. We have about 22 million people living along the Southern California coastline, and every year we get about $14 billion in revenues from visitors coming to our beaches. So we want to understand how to keep them clean and how future changes might affect them, because we need to be able to predict how we can also adapt to keep up with that global change. So the first study I'm going to talk to you about looks at the way that stratification changes in the future might affect phytoplankton growth in California. And this was a study I did in Northern California. And there's a process called upwelling that we have along California. Just as show of hands, who's heard of upwelling before? Californians. It's great. So upwelling is basically this process where you have this warm, you have stratification, just like in your cups. You have a warm layer that's very nutrient poor on the top, and then the deep water is colder and full of nutrients. The reason it's full of nutrients is because as cells die, they sink down and then they get regenerated, and all the nutrients that were in their cell gets released into that water. So now that water has high amounts of nutrients in it, like nitrate. And so when winds blow these waters offshore, the deep water has to rush up to take its place, and that's what upwelling is. It brings those nutrients with it, and as soon as it gets into the sunlit layers of the ocean, the phytoplankton can take it up and make a bloom. Now, there have been a couple climate simulations to try to figure out how the ocean might change in the future with global warming, because we expect that stratification is going to become even more intense. Less mixing in the open Pacific Ocean, less ventilation is what we call that. As a result, because there's less mixing, more and more nitrate will build up in the deep water. So we can expect that by the year 2100 in our coastal waters, when upwelling occurs, a lot more nitrate is going to come up with it. And that will almost certainly affect algal blooms, but the question is how? How is it going to change our species in California? California is kind of a tricky place because even though it has very high nutrients from upwelling, it also has co-limitation for different nutrients. That means that more than one nutrient can limit phytoplankton growth. So in this case, there's a micronutrient called iron that's very important. And if you look at this study site, what should jump out at you here is that there's this region down here where the continental shelf is very, very shallow, and it's very, very narrow. And those little places, that means that there's not a lot of suspended sediment coming off of the ground and going into the water column. So that site tends to be iron limited because the iron comes from the earth. But if you go a little bit farther north, you have this broad shelf, and all of that current sweeps sediment up, and then there's plenty of iron in the water, and so the phytoplankton tend to be nitrogen limited at these sites. But in the future, if we start adding more and more nitrogen, what's going to happen to these sites down here in the bottom, which are right now are iron limited? So to answer this question, I did a series of experiments at 5 different sites all around Monterey Bay and San Francisco Bay, and each of them are color-coded. That'll continue consistently throughout the talk. But when I do these experiments, what I do is I take a boat out to a station that's, not being directly impacted by any effluent or something like that. Collect it, disperse it into these bottles, and then I can add different chemicals to the bottles and monitor how the phytoplankton change over a period of several days. And that way I can tell what they're hungry for. If I give them nitrate and they grow a lot, then I know they were hungry for nitrate. If I give them iron and they don't grow, then again, I know they weren't hungry for iron. So these plots are showing chlorophyll concentration over the course of the experiment at each of the sites. And the triangles are showing you everywhere that I added nitrogen. And what you can see is that 3 of the sites that I tested were nitrogen limited despite having incredibly high values of nitrate just naturally from upwelling. But the nitrogen that I added simulated what the situation is going to be like in the year 2100 when we have that excessive nitrate from upwelling. To get a closer look at which members of the community respond, I did microscopy to count different types of cells. And this is a type of cell over here on the right that you guys might have heard of. It's called Pseudonychia. This is the one that's been causing these massive blooms all along coastal California. And it causes seabird mortality, it causes mortality in marine mammals, and it also poisons shellfish, making them inedible to people, because if we eat them, then we can get sick as well. So it's a major economic problem for us in California. But what I found was that in samples where I added nitrogen, I actually found less of these toxin-forming cells. And instead of these cells, I tended to find more single-celled things that didn't form chains. So here on this plot, it's showing the percent of chain-forming organisms right here for each of the different sites. And what should jump out is for anywhere that I added nitrogen, which you can see with the N, there was a decrease in the number of chain-forming cells. So something about that nutrient availability affects whether they form chains like this or they exist as single cells. You can think of it like beads on a necklace. You can have individual beads or you can have the beads together on a necklace. When you give them plenty of nitrogen, you just get single beads. And when they're starved for nitrogen, they make a necklace. The reason that that matters is that it affects sinking rates of cells. So things that are bigger and things that are jumbled together are more likely to sink. And when that carbon sinks into the bottom of the ocean, it can be sequestered for long periods of time, and that, of course, affects climate because it's removing CO2 from the system. So with the nitrogen additions, we tend to see more of these single cells. In contrast, in the other treatments where I added iron and they retained that iron, that nitrogen-limited characteristic, we got beautiful blooms of chain-forming cells. So these are diatom cells called Thalassiocyra, which is a really common species that we have off of our coast. And you can see the little filaments that hold it together in the necklace. To try to understand how geographically these patterns could change in the future under the simulations for the year 2100, I used existing nitrogen and iron data for each of the sites. And they range from about Monterey Bay in the south to Point Reyes in the north. And this plot is showing you, see that green shaded region? Everything to the right of that are regions that are iron replete. That means that based on the iron to nitrogen ratio, the cells probably have enough iron to grow. And then everything that's kind of in the shaded region is transitioning. It's kind of on the cusp. It could really go either way. And then if it's to the left, it means it's iron limited. What each of these blocks shows you is on the right-hand side, the modern ratio, and on the left-hand side, what the ratio will be by the end of this century if the changes in nitrate occur like the model says they will. And so what you can see is that there are certain areas down in Monterey Bay and a couple further north that are currently iron replete, but that start to transition towards iron limitation as we start to add more nitrate. And then there's others which are currently on the cusp, which are pushed into full-bore iron limitation in the future. Now remember, the reason we care about this is because it changes the way that they bloom and the way that they form their cells and whether that carbon's going to sink. So we know this bodes for changes in the carbon cycle. So the main take home of this study was that, yes, if you add more nitrate, the cells respond. We tend to get more free-floating cells that are single. They don't sink as much. That could mean more food is available for the little animals and the smaller fish that are living in the euphotic zone with the algae, but we don't really know how that will trickle up the food web. Because if we're changing the size of organisms that are at the base of the food web, we're favoring different grazers, and that in turn could potentially favor different types of fish. So these are ecological changes that need to be investigated. Additionally, a lot of the studies on harmful algal blooms have predicted that future warming events, warming of the ocean, will cause more harmful algal blooms. But what this study really suggests is that there's a feedback in the nutrient cycle that might circumvent that from happening. So instead of seeing more pseudonychia blooms in the future due to temperature, we may actually see less because of the stratification and the reduction in nutrient inventories. So I also wanted to just introduce really briefly a new study that I've started since coming here to UCI that my team has been working on. And it's really looking at what happens when our coastal ocean starts to resemble the open ocean. So this is a false color image showing chlorophyll distribution patterns where the warmer colors are like high chlorophyll and the cooler colors are very low. And what you can see is that in the open ocean, we have very, very low chlorophyll. And what we tend to get out there are these tiny little cells called prochlorococcus. It's the most abundant photosynthetic organism on the planet. And they're tiny. They're about 1 micron wide. So if you put 100 of them side by side, it would be about as wide as one piece of human hair. They're very, very tiny. But they do a lot of photosynthesis, and we rely on them. We don't tend to find them in coastal waters until recently. A colleague of mine in the Earth System Science Department named Adam Martini has been doing a field sampling campaign for the last 3 years, once a week at Newport Pier, right down the street. And what he found was that there's been this warming trend that's occurred over that 3-year period, and that with that warming trend, we tend to have more prochlorococcus, which makes sense because prochlorococcus is from the warm open ocean. But the question is, we see it coming back every August. We don't know if it's a natural native population that just blooms at the end of the summer, or if it's just getting kind of coming in with the currents, like it's not really meant to be there, it just shows up because the water comes and brings it. So what my team has been doing is increasing the frequency of those sampling events. So we go out 3 times a week now to sample at Newport Pier to try to understand what's driving this. And we're using actually the El Niño that's happening this year to help us really get a good feeling for what these prochlorococcus cells are like and why they're in our coastal waters. The way that the El Niño works is that during a normal year, we have these trade winds that are blowing offshore, and that tends to push a lot of our warm surface water that's nutrient poor over to the western part of the Pacific. That's why you have this warm water, all this evaporation, lots of rain over there. We're Mediterranean, we don't get so much rain because we don't have that warm water. During a La Niña, we have intensification of those winds. And so an extreme amount of warm water gets pulled up in the West Pacific. During those years, we're exceptionally dry because we have even less humidity in our air than normal. But what happens during an El Niño is exactly the opposite, and this is what's happening this year. We have relaxation of these winds, and what that allows is that water that's been built up in the Western Pacific comes slamming into our coastline. And it's not dense, and it's warm, and it's nutrient poor, so it looks an awful lot like the open ocean. And it sits right on our coastline, and it prevents upwelling from happening. So it changes our nutrient patterns. So we expect that because this is a strong El Niño this year, even though we have not gotten the rain, that's for sure, but we have been seeing changes in the ocean patterns that we think will make it more suitable for prochlorococcus. So our aim is to do some single-cell genomic analysis where we can actually take members of this population and sort them into individual test tubes and sequence their entire genome from a single cell. This is brand new technology that's only been available for the last maybe 10 years really on these marine phytoplankton. We're very excited to be able to compare the genomes that we get from this coastal population to genomes that we already have for the open ocean ones. Because then we can start to ask questions like, what genetic adaptations do these cells have that make them better fit to living in our coastal waters? For example, do they have a broader temperature tolerance, or are they able to use more nutrients that are common to the coast that they wouldn't find in the open ocean? So to the next part of the talk, I wanted to give a nod to the fact that not everybody is excited about phytoplankton as I am, and that there are other reasons why you might care about the ocean. My grandfather was a good example of that. He was the first person in his family to be born in the United States. His family came over from Finland, and he loved to crab on the Chesapeake Bay. And so when I was a little girl, one of my first exposures to the ocean was going crabbing with him every year. So there's a lot of people in the world who really have emotional and spiritual ties to coastal waters, and their livelihoods rely on it. So in this next study, I'd like to share with you an example of a region where this type of global change has been happening at an incredibly fast pace. And we can actually measure these changes happening, and it's affecting people's livelihoods via the fisheries. The East China Sea and Chinese coastal waters are the largest fishery in the world. They support about 9 million jobs and produce over 57 million tons of food for people every year. So they're very important economically and socially and culturally. What they've been having is an increase in harmful algal blooms that have been occurring for about the last 3 decades. Back in the 80s, there was less than 10 harmful algal blooms in the East China Sea per year. And up to about 2003, now they're having almost 100 per year of these harmful algal blooms. And you can see that they're pretty intense. They're changing too. This is a dinoflagellate bloom, and I'll talk to you about them in a second. But that's a different type of algae than used to grow in the East China Sea. They're seeing it a lot more commonly now, and it's not a normal native population. Normally they would be having blooms of diatoms in the spring, similar to what we have here, but instead they're getting lots of dinoflagellates. So I was interested in understanding why. Some people have predicted or suggested that changes in the effluent from the Yangtze River is the cause of it. And the reason for that is that a lot of the nitrogen that's being applied in agriculture eventually makes its way into the river, and so the river water is a huge source of nitrogen, and it fertilizes species that wouldn't otherwise be fertilized in the coastal waters. But more work has been done recently on atmospheric deposition of dust and pollutants and how that could be enriching different phytoplankton. And so that's what this study is about. So I wanted to introduce to you the idea of atmospheric deposition, because it's not something that most people think about on a day-to-day basis. This plot is showing you the average annual deposition of dust, natural dust that comes off of deserts, comes off of land, and it goes over into the ocean. And all of that dust contains nutrients that the phytoplankton need to live. So just like you might go to the store and buy some Miracle-Gro for your garden to get the nitrogen and the phosphorus and the iron, the natural Miracle-Gro for the ocean is in the form of this dust. So you can see a huge source is the Sahara Desert. It's very arid, so when the winds pick up, it blows all that dust into the ocean, and it fertilizes the algae. Everywhere you see a star blinking on there is a place where I've gone and experimented with the algae and measured their responses to dust. And today I'll talk to you about the experiment in China. So dust is natural stuff, crustal material, but it can also include stuff like ash from fires, which has a lot of nitrogen in it. It can include biomass burning, emissions from cars, emissions from factories, biological particles, and even sea spray. The ones that I want to talk to you about today are these anthropogenic or human-caused sources of aerosols, because they tend to have not only more nutrients and metals in them, but they're more bioavailable, they're more soluble. The organisms can get at them better. So they don't just sink away, they actually get used. And this is an image of the East China Sea from where I collected the water. And it looks just kind of like a hazy, overcast day, but it's not. That's actually the pollution in Shanghai. This is what it looks like every day. And so during the whole time that I was there, I never saw a blue sky. All of this is coming from anthropogenic sources of combustion, and it goes out over the ocean, and it settles down on the surface of the ocean where it can affect the phytoplankton. So to try to understand how these factors might be influencing the phytoplankton, we took a two-pronged approach. The first was to use a satellite record to see if we could match up growth of the algae with big deposition events. So there's a satellite called Modus Aqua that is really good at measuring aerosols and chlorophyll at the same time. We used land records or ship-based records to tell us when there were blooms of diatoms versus dinoflagellates, so we could parse the record even further into blooms that we knew were dominated by one species or the other. And we looked at the onshore site, which was close to the Yangtze River. Remember, that's getting a lot of nutrients from the river. And then an offshore site, which should have been more removed from the river mouth. And certainly what we saw when we were trying to figure out whether the aerosols were causing blooms of phytoplankton, in the coastal water, there really was not that much of a response. But offshore, we did see it was about a 5-week lag. So aerosols accumulate over a couple-week period, and all those nutrients continue to add to that bloom and fuel it, and then by the end of that 5 weeks, you're in a full-blown algal bloom. So we do know that there's a relationship between the deposition and the algae. So to try to get an even closer handle on this in terms of which cells were responding and how fast they were growing, we did another incubation experiment, similar to the one that I told you about in the prior study. And it was conducted in the East China Sea. There were 2 sites. So right offshore of the Yangtze River Delta, there's a little archipelago right here. We collected dust samples at these islands. So basically just a big pump that pumps dust onto a filter. And then we can add those different types of dust to water that we collect either from an onshore site, which was within the plume of the river, or from an offshore site. And what we saw was that, just like the satellite record suggested, for the onshore site, there's really not that much of a difference when we grow out the algae. They just tend to all grow. And the reason for that is that if you look at this outflow of the Yangtze River, see how much sediment is churned up there? So these algae have a ton of nutrients in their water, but they can't use it because they're light limited. So when we put them in our bottles, everything grew the same way because they already had plenty of nutrients. They weren't nutrient limited, they were light limited. The story was really different when we went offshore, though, because offshore the sediment has kind of settled out, and now they have plenty of light and we can start to work on the nutrients. So when we add nutrients like nitrogen, phosphorus, or NNP together, we see a really strong growth response. These samples right here are all the different types of dust that we added. Each of them has a different chemical composition, different metals, different amounts of nitrogen. Some of them caused a positive growth response and others didn't. So to get a better idea for who was growing and who was actually responding, I did more microscopy, similar to the other study. Here are all the diatoms. What we can see and what you can jump out at you is that for the most part, if you add aerosols, you do get a slight increase in the diatom cells. And using principal component analysis, we can put in the concentrations of metals and nutrients and determine what combination of factors these cells respond to. And it seems that they really like it when there's a normal amount of trace metals like iron being added and slight enrichment from nitrogen. Again, just like in the last study, the exception was the pseudonychia, which forms that domoic acid toxin. So they have that in China as well. And what I saw was that they responded negatively to any of the nutrient additions or the dust. So all of this increased dust deposition on these types of algae actually does not favor them. What was really interesting about this was these dinoflagellates that I told you were sort of taking over this region. Just dinoflagellate, if you're familiar with the word, dino and what that means, you think about dinosaur, it means terrible lizard. Dinoflagellate means terrible flagellate, and you can see from the way that they look like little monsters, but they're really beautiful under the microscope. Every time you find one, it's just like, Christmas morning. But they respond really strongly to aerosols. Look at how well they do. And so in the principal component analysis, what we were able to tell is what's favoring them is actually an increase in iron and a decrease in phosphorus. So they're actually not responding to nitrogen fertilization. They're responding to the low phosphorus values that the system drives itself towards. So you add a lot of nitrogen, eventually you use up all the phosphate. They're better at dealing with that starvation than all the other ones, and that's why they're able to outcompete. So that's why we're seeing more blooms of it. So this study was really able to show at the end of the day that this long time scale, 30 years or so, shift in the regime of phytoplankton blooms in the East China Sea could be due to these anthropogenic aerosols. Another interesting thing about dinoflagellates, just because they're fascinating, is that they can eat other cells. So that's another way that they can maintain their blooms even once they've drawn down the nutrients. So if you look down here where I have arrows, see these little circles? That cell actually will split in the middle like Pac-Man, and it'll eat another cell. So it has chlorophyll in it, it can do photosynthesis, but then when it runs out of nutrients, it can just eat its neighbor. They're crazy. They're like half animal, half plant. So what this study's goal was to try to understand the ecological changes that are occurring as a result of the physical and chemical changes in the East China Sea. So we know that aerosol nutrients and trace metals can alter the seawater chemistry, but the effect that they have on the algae is really different. It depends on the species of algae, and it depends on the proximity to other nutrient sources like the Yangtze River, so there's a geographic component as well. So I'm going to wrap up the talk now, but I just wanted to end kind of on a positive note, because sometimes, I don't know, when I talk about climate change and the way that the ocean is responding to pollution, it can be kind of a downer. And our department is really focused on understanding the way that the Earth changes over human lifespans and what our role in that is. And implicit in that, I think, is the fact that if we are causing the problems, we can also be the solvers of the problems. And after all, we know that UCI, has been a leader in a lot of these different types of fields. People can really get behind the idea of developing new green technologies, harnessing the power of the free market to make that happen, getting behind values that allow us to leave nature pure and pristine so that we don't leave our kids with problems that are too big to be solved. I take a lot of inspiration from some of the early founders of people in the School of Physical Sciences. And one thing I wanted to mention before I close was that I had an opportunity to talk to several of the students and colleagues of some of these people, like Sherry Rowland and Norm Rostoker, at a symposium last summer. And I was really surprised by how inspiring and how similar their stories are despite being in different fields. So I never had a chance to meet these people, but I know some of you in the audience have. So if you would like to read this paper, it has some good anecdotes and stories about these gentlemen, please feel free to contact me and I'd be happy to share that with you as well. Okay, so with that, I'd be happy to take any questions that you might have.